It's a great pleasure to introduce our plenary speaker, Professor Ken Taylor from University of Colorado. He's actually a clinical neurologist working with several viruses causing neurological infections, and I think he has done a lot of clinical work, but also absolutely amazing work trying to understand pathogenesis of them. And he's here today to explaining us what we understand of this new polio infection caused by enterovirus D68. And I think most of you might be aware of his really groundbreaking work on developing mice models and trying to see what has happened and why we are seeing this infection just now. So very great to have you here, and floor is yours.
Thank you very much. I want to thank the organizers and our co-chairs who've done a lot of work, of course, on enterovirus surveillance and been part of our understanding of enterovirus D68 and the acute flaccid myelitis story. I'm going to try to tell this to you a little bit from an American perspective, and we'll talk a little bit about some of the European features, hopefully, as well. No story like this is a one-person story, so I want to start by introducing some of the people I had the pleasure of working with in these studies. This woman here, Terry Schreiner, in the corner, is a pediatric neurologist in my department, and she called me up one day in the summer of 2014 and asked me if I wanted to walk across our campus at the University of Colorado to the Children's Hospital to see some unusual cases of a polio-like illness that they were seeing. Kevin Mezikar, right above her, is a pediatric infectious disease specialist. I'm the co-mentor with Sam Dominguez, who's in the other corner there, of his NIH K Award. And as many of you know, he's been a leading figure from the pediatric infectious disease side in studying AFM and its epidemiology. In the middle there, Dan Pastula is another member of my department who previously worked and now currently works a little bit with the Center for Disease Control in the United States and was responsible for studying parts of the early epidemic. And a lot of the basic science work that I'm going to tell you about in both the mouse model and in cultured neurons was due to Allison Hickson, who was an MD PhD student doing her PhD in my laboratory and now back being a medical student at the University of Colorado. And we've had a lot of collaborators at UCSF and at the Venter Institute and elsewhere. So the story I'm going to tell you, and I'm going to try to tell it a little bit as a story, began in the summer of 2014 in Denver. As I told you, the first case, almost the index case for us that's drawn a lot of attention to this outbreak, occurred that summer at the Children's Hospital. And very quickly it was clear that this wasn't a unique patient and that we had had a very unexpected cluster of 9 cases within less than a month. And this caused us to report this to our CDC, who quickly issued a national health advisory, and a very quick early paper came out in Morbidity and Mortality Weekly Reports that you can see there on an acute neurologic illness of unknown etiology in children in Colorado. And this occurred in a very unusual summer. We were in the midst of a surprising outbreak of respiratory infections requiring admission to the hospital in young children. Sorry, it's a little awkward for me because of the angles here, but if you look at the sort of green line with the squares that runs across there, that was sort of about the typical number of weekly admissions, around 20 or so, that we would have to our children's hospital over the weeks shown across the bottom in a typical season. And then the red mountain peak that you can see, that you can go back, that you can see above that was about a 3-fold spike in the incidence of these respiratory admissions to the hospital. So everybody was sort of overwhelmed. And what I want you to notice is underneath the mountain peak, those purple bars, this was the coincident appearance of these cases that we would now call acute flaccid myelitis that were happening time-linked to this unusual surge in respiratory admissions. Oops, go the other way. Very quickly and over time, the case definition for this syndrome of acute flaccid myelitis has been modified, updated, and changed. This is the most recent one from what we call our Council of State and Territorial Epidemiologists. The CDC and others make suggestions. They come up with rules in the United States. These are the rules that states use for describing and reporting illnesses. The CDC, when it confirms cases, has a panel of expert neurologists. Dan Pastula serves on that panel, Jim Sejvar and others, that look at the data for the cases and decide whether they're confirmed or not. This is a syndrome definition, meaning a running together of a group of signs, symptoms, and laboratory features. And the key ones are the acute onset, as you can see there, of flaccid limb weakness associated with laboratory evidence on magnetic resonance imaging, or MRI as we call it, of a spinal cord lesion, mostly in the gray matter where the neurons are, typically more in the anterior horns where the motor neurons are, and spanning more than one segment. Most of the patients will have a CSF pleocytosis as well, and if you meet all of those criteria and it's validated, you're considered a case of AFM, or acute flaccid myelitis. Whenever you come up with a syndrome definition, there's always a lot of backfighting about whether you have a big tent that encompasses a lot of different things but captures everybody of interest, or a more narrowly focused, smaller tent that has the risk of losing some things that aren't typical but is more focused. This is sort of a big tent. These are pretty vague things, so it is not surprising that you are not going to find just one cause for all of the cases. So don't let that confuse you. Oops. Get my directions pretty good. This is the latest map from about a week ago of the CDC's accounting in the United States of the cases in the last 2018 season. Every state that's colored in blue there reported cases of AFM, so you can see there were a few like Alaska that did not. Colorado, where I'm from, is sort of in the I don't have a very good pointer here, but sort of in the middle right there with 16 cases, which I think was second only to Texas, and the total was about 235 cases last year. It follows a very interesting and unusual epidemiologic pattern, which is this biannual, every other year, set of spikes in the number of cases with an intervening quiet period. So you can see there were, in that first year that I was just telling you about 120 confirmed cases by the CDC in 2014. It jumped to 149 in 2016. It's over 235 confirmed so far for 2018. You can see underneath the bar graph I just sort of wrote for you the number of states. So not only has the number of cases increased in each of the spikes slightly, but the number of states reporting cases has jumped from 34 to 39 to 41 of our 50 states as the time has moved. And you see the rough seasonal incidence. So in the US, it's sort of, right around now, a late summer, early fall pattern. It's probably easiest if you just look at the far column there where it says total. This is the clinical features roughly of the cases that we've seen as collected by the CDC and others for nearly 440 cases. You can see this is predominantly a disease of children. The original case definitions, in fact, required that you be under the age of 18. That's subsequently been dropped, but even if you drop that exclusion, about 90% of cases are pediatrics. And by the way, the adult cases are probably something else, including West Nile, and different than the pediatric cases. There's a slight male predilection, and we've seen it across a variety of different racial groups. Almost all of the patients have a preceding viral-like prodrome. It's about 90%, typically respiratory or febrile. You can see about 98% will have one or the other of those two, and a little less commonly a gastrointestinal prodrome. The prodrome typically precedes the onset of neurologic disease by several days. You can see the febrile illness by a median of about 2 days, and the respiratory illness a median of about 5 days. The neurologic features of the weakness are kind of summarized in below the little thing I wrote there below the table. So you have the acute onset of a flaccid hyporeflexic weakness, decreased tone in the limbs. It is usually asymmetric. It is a little more proximal than distal, meaning the upper parts of muscle groups in the extremities or in the legs versus the feet or the hands. And it is typically worse in the upper extremities than in the lower extremities, and sensation is usually preserved. And there's a little more information, some variability across each of those columns was a different hospital series collected at different times, but again, asymmetric weakness, flaccid, usually upper more than lower extremities, and with preserved sensation. The spinal fluid is almost always abnormal, showing a pleocytosis. I think on this chart here, says about 81% of the cases had more than 5 cells. The quicker after the onset of paralysis you look at the spinal fluid, the higher that number tends to be. The predominant cells are lymphocytes. The protein is often a little bit, but not dramatically elevated in most cases, and the sugar is normal. So a typical kind of viral-like prodrome of lymphocytic pleocytosis, elevated protein, normal sugar. The imaging findings are very, very characteristic, and you don't need to be a neurologist or a neuroradiologist to usually appreciate them. You can see this patient here has this tremendously long multi-segmental area of increased signal in the middle of his spinal cord. In the longitudinal sections, these are more like bread slices or cross-axial sections, and all of that abnormality shouldn't be there. And this is a little bit later from a patient where you almost see those like owl eye like increased areas of signal right in the anterior horns of the spinal cord. Occasionally we will see signal outside of the spinal cord as well. And I'm going to jump backwards to this in a second, but if you look at the subset of these patients who have illness linked to enterovirus D68, it is a bad thing to have. It is a polio-like illness with some nuanced differences. If I was talking about polio, I might have told you lower extremities more than upper extremities, for example, but the basic pattern and type of weakness, the pleocytosis, these other things are very polio-like. The children do have some recovery, but in those where it's been linked to enterovirus D68, it tends to be modest, usually in the first 3 months. This is from a paper we wrote in neurology, looking at some of the first cases we saw in 6 months follow-up. This is a young boy in panel C up there, and he's being asked to raise both his arms in the air, and I think you can see how much difficulty he's having raising his left arm. In the lower panel, this was a little boy who had been affected at about 18 months, a year and a half of age. This photograph was at about 2 years of age, and I think you can see the withered and atrophic right leg there. Again, the enterovirus D68 cases have tended to have a severe paralysis with only modest recovery. This is probably the oldest child I saw in that first summer DH. He's now in his 20s, and we've continued to follow him. Again, this is about 8 months after the onset. He was a high school senior in the United States when he was affected, and you can see again the tremendous degree of atrophy in the shoulder muscles, in the arms, across the chest. He even has a feeding gastrostomy tube that you can see in place there. This is not a good illness. So everything I've pretty much told you up to now, I think, has been the subject of pretty much general agreement. Of course, one of the mysterious questions has been what is causing this. I think the evidence now has accumulated dramatically, despite earlier controversies, to say that this disease, those biennial spikes, the severe cases that I'm telling you about, is closely linked to enterovirus D68 in the majority of cases for which we have an identified etiology. We had a little outbreak in Colorado associated with another enterovirus, A71, but clinically the disease looks very different, and we could talk about that in the questions if people want to. About half of the cases, as defined, as I showed you on that earlier slide, will have amplification of enterovirus D68 by RT-PCR from oropharyngeal or nasopharyngeal swabs. And that and the epidemiology was one of the first, are these two things causally linked? Metagenomic next-generation sequencing of spinal fluid has not tended to reveal any significant pathogens, and that should remind me to tell you that if you do CSF PCR, RT-PCR for enteroviruses, you also typically do not find any positives. There have been isolated cases, less than 1%, where you do, including enterovirus D68. This has been done by multiple groups, Charles Chu and others at UCSF, Ian Lipkin at Columbia, among others. One of the more exciting developments in the field in terms of etiology has occurred in the last couple of months, meaning July and August, where two groups, one at UCSF, including Mike Wilson, Charles Chu, and others, and one at Columbia, led by Ian Lipkin, have used large-scale peptide arrays to look for antibodies against viruses or enteroviruses or specific enteroviruses in CSF. And both of the two groups, Ian's paper is just out in MBio, and the UCSF paper out now in a preprint server, bioRxiv, I put the references at the bottom there, have clearly shown an increased prevalence of antibodies against enteroviruses generally and against enterovirus D68 more specifically, and including identifying specific regions on VP1 and other proteins that antibodies have been directed. This so far is the closest thing I think we have to a smoking gun linking enterovirus D68 especially to this disease. One of the reasons that the positive rate for isolation of specimens has perhaps been a little lower than some of us might have thought it would be is undoubtedly a timing issue. If you look at oropharyngeal or nasopharyngeal RT-PCRs to amplify enterovirus D68, if you don't get it fast, you don't get it. So if you just look at where the blue colors are out of the total bars there, the top graph is in relationship to the onset of febrile illness, the bottom to the onset of limb weakness, and you can see that very quickly those become negative. So if you look at early, you get much higher incidences than if you look late. When these cases appeared, and I went over to the Children's Hospital in the summer of 2014, that graduate student that I mentioned to you, Allison Hickson, had just come to my lab. And those of you that run a lab may excuse me for saying this, but she sort of showed up and I had no idea she was actually coming. I had totally forgotten, had put exactly 0 seconds of thought into what her project was going to be. And because when she showed showed up and said, "Hi, I'm Allison Hickson, and I'm here to do my rotation," I had just literally walked back from Children's Hospital. I, with no thought and no idea of how easy or hard it was going to be, said to her, "Hey, why don't we try to see if we can make a mouse model of this weird polio-like disease that we're seeing at Children's?" And she, of course, just having arrived at the lab and not knowing what the struggles were to do just that very thing for polio and how difficult it was, said, "Oh yeah, sure, what a great project." For what that's worth, we collected a bunch of isolates from the 2014 season belonging to different viral clades. I won't go into the epidemiology, but enterovirus D68 can be separated by molecular phylogeny into a variety of clades and subclades. The original viruses that were the prototypes for this group are viruses referred to as Furman and Rin. Those, interestingly, isolated in the US and California in the 1960s, haven't been linked to paralytic disease in animals or in people. When we stuck these into mice, we very quickly discovered that many but not all of the strains isolated in 2014 produced paralytic disease in mice, just looking at the upward bars there. The original prototype strains did not. The one tiny little black bar there for Rin was probably due to the myositis it produced, not to a neuroparalytic disease. So without doing anything special to the mice, neonatal mice, Swiss Webster, we stuck the virus originally intracerebrally because that's the way you get neurologic disease. If you want to, you use baby mice, you stick it in the brain. Without any sort of transgenics or anything like that, we were seeing paralytic disease. The disease appears, if you look at the red line, dotted line in the upper curve there, pretty quickly, about 3 or 4 days after, again, we switched later to an intramuscular model of inoculation. You start to get paralysis that peaks and plateaus within a week or so. We quickly realized that the paralysis was associated, as seen in the lower graphs there, with, especially where it says spinal cord, with rapid growth of virus in spinal cord. There was a dose-dependent appearance of the paralysis. So by changing the inoculum of the virus, and we did this across several strains, just one of which is shown there, we could get more severe and more acute paralysis as a function of dose. And we quickly learned when we were doing this in Swiss Webster mice that the route of inoculation seemed to be critical in the incidence of the paralysis. So if you stuck it into a muscle, you could get 100% paralysis. This is just with one of the strains that we used. The brain dropped that down to about 50%. And if you tried intraperitoneal or intranasal, you got disease, but it was sort of unusual, down in the single-digit percentages. And those of you that are really good at remembering may remember when this epidemic appeared in 2014, there was a huge increased ratio in the number of patients with enterovirus D68 respiratory disease compared to paralytic disease. So probably these low incidences are very reminiscent of the human condition. This has been replicated now by numerous other groups with slightly different models. There are 2 groups in China. There's a group in Utah that have been doing now mouse models for this. The Chinese group had an interesting observation that they can get disease with higher frequency with some of these routes of inoculation that may depend on the mouse strain. So that may be an interesting clue to us that there are host factors. If you use a BALB/C mouse or an ICR mouse, very susceptible. C57 black sort of in the middle. And we stupidly, we didn't know any better, but they were cheap, picked Swiss Webster, which is probably the least susceptible, but gets disease quite clearly, as I think I've shown you. There's an age dependence to the paralysis in the mice just like there is in the people. So if you look at those family of curves on the top there, you can see the motor impairment scores. So higher up on the curves is worse. As we change from very young mice at day 1 all the way up to about 3-week-old mice, the incidence of paralysis declines. And there's a great correlation between paralysis and the ability of the virus to reach and grow in the spinal cord. Maybe I'll play you the 2 movies. He's going to have to do it for me. This poor guy has a little left forepaw weakness, so you saw him scattering it around. The black marks are to identify him. They're not part of the disease. The more striking one is the guy in the lower guy here. He's paraplegic, and you can see him dragging his lower limbs. This is at about 12 days after infection with the virus. So we get all the same types of paralysis that you see in humans. As I alluded to, this is from an original paper in PLOS Pathogens. Virus grows either by titering infectious virus in the spinal cord, TCID50, or by RT-PCR in the spinal cord. There's a beautiful neuropathologic correlation between where the animals are paralyzed and where you see death, loss of motor neurons, and viral antigen. So this little guy had a left hind limb paralysis. If you use choline acetyltransferase to stain for motor neurons, you can see there with the side with the white arrow above it that in contrast to the other side, there's loss of the choline acetyltransferase positive motor neurons. If you catch these animals at the right time before, of course, all the motor neurons are gone, you can detect enterovirus D68 antigen. Here staining, I think this was for the VP2 protein, just showing it to you a little clearer and a higher magnification there. So infectious virus in the spinal cord, viral nucleic acid in the spinal cord, loss of motor neurons corresponding to the paralysis and viral antigen. If you do electron microscopy and you look at the motor neurons, they're jammed packed with enterovirus equivalent or consistent particles. So you can see as you move there from 11,000 to 49,000 to almost 100,000 magnification, those viral factories and particles inside the motor neurons, and just for a comparison, a reference of what enterovirus D68 looks like from the CDC collection. Of course, doing this in mice made us interested in would these grow in different types of motor neuron-like cells. We started in collaboration with David Brown and Richard Scheuermann at the J. Craig Venter Institute using a line called SHSY5Y cells, but start at the bottom panel where it says HeLa. Our neuroparalytic strains and the ancestral non-neuroparalytic strains all happily grow in HeLa cells. If you just look at the middle row there with the red staining, you can see happy growth of all the strains in the HeLa cells. But if you jump up to the top panel and look now at the SHSY5Y, only that Missouri 47 strain shows you the red antigen. It's not seen with the Tennessee, the 197 strain, or the mock infection. So when we did this with more strains, it did seem like there was a very nice correlation between the ability of strains to cause paralysis in mice and to grow in a neuronal-like cell line. Same thing as I showed you for antigen. I won't dwell on this for a CPE, cytopathic effect. So HeLa cells, all of the strains except the first one where it says mock, destroy the monolayer fairly well, but if you'll notice, only the Missouri strain shown in red produces cytopathic effect in the SHSY5Y cells. There's a difference in the receptor utilization of these strains in neuronal cells. Now this is human iPSC motor neuron-like cells, and it turns out that the old non-paralytic strains seem to be very dependent on sialic acid. So if you treat as a receptor, so if you treat cells with neuraminidase, you abrogate much of the infection and binding of the old strains, but not of the new neurotropic strains. They seem to be able to infect motor neurons in a sialic acid independent manner. We've also looked at the ability of neurons to transport viruses, and you can use clever culture systems, including this one that I've drawn out here. This is from a paper that was just out last month in the Journal of Virology. In these multi-chamber slides, you can put motor neurons or human iPSC motor neuron-like cells in one chamber. Their axons grow across a series of microgrooves into a second set of chambers and enables you to infect at the cell body side or the distal axonal side and study infection. So you can see the cell bodies, the grooves, and the connection. And what you see is if you infect at the periphery of those cells in that axonal chamber, the virus happily spreads to the cell body of the motor neurons, happily grows. So the green staining, again, just a little hard with the pointer, but the green is viral antigen. The microgrooves would be at this end of the slide. So we sort of infect where this panel is. Virus spreads across the microgrooves. There's viral antigen. There's viral growth. If you chemically, mechanically, or otherwise inhibit, sever the axons, mechanically damage them, or inhibit axonal transport in them, as shown in the lower graphs here, you dramatically inhibit the spread of virus. It is transsynaptically spread so that you can literally follow in these chamber systems. Here's infected neurons, here's a second generation, there's a third generation, and they are the ones that are connected together. Interestingly, the spread seems to be almost exclusively, with all the strains we've tested so far, retrograde axonal transport. If you infect the cell body and put some indicator cells where the distal axons are to see if they'll get infected, you do not see that. So some of these unusual patterns of spread, including why intramuscular injection in a mouse may be a great way of doing this, undoubtedly, I think, have something to do with the patterns of axonal transport and this favoring of retrograde versus anterograde transport. If you have a mouse model like this, you can use it to potentially test therapeutics. I don't know, I suspect it's pretty identical. When this appeared in the United States, we sort of tried everything we could possibly think of, from available antivirals through steroids, through intravenous immunoglobulin, often given simultaneously in, of course, a non-controlled fashion. There was no clear rationale or ability to look at efficacy. I'll tell you, in a mouse model, if you give human intravenous immunoglobulin, and in the US, our lots of intravenous immunoglobulin contain fairly high titer neutralizing antibody against enterovirus D68, you find clear protection against paralysis in the mouse model. So being higher up on that curve is worse. So there's the saline control at the top. And if you give IVIG at the same time as virus, day 0, or a day later, or 2 days later, you get almost no paralytic disease. If you delay it out to 4 days, you sort of get an intermediate phenotype. I actually think I put an older slide in here. I could have drawn a line right almost between those two for day 6. And again, to remind you, in the mouse model with an intramuscular inoculation, paralysis begins at about day 3. So even after the onset of clinical symptoms, if you're a mouse, boy, do I have a potentially good treatment for you. So IVIG seems to work in the mouse model. Protection correlates with inhibition of spread to the spinal cord. So if you just look here, you can see these are the IVIG-treated animals. Here's the muscle titer dropping down to virtually non-detectable. And in the far panel, the C panel there, same thing in the spinal cord. We got very interested in trying a drug called fluoxetine, which is widely used as an antidepressant, including in children, because there was some in vitro data suggesting that fluoxetine, having nothing to do with its antidepressant efficacy, having nothing to do with its mechanism of action in depression, also was able to inhibit the viral 2C protease in vitro. In the mice, once we tested this, it really had minimal, if any, effect, so a very different result than IVIG. You can see not much on the motor impairment scores, not much in terms of the impact on growth. So it didn't help, but it didn't really hurt. Steroids were frequently used in these patients, often as dexamethasone, but in other formulations as well. And here we saw a third pattern, an actual worsening of disease if animals were treated with steroids. So in most of the models that we were using for our therapeutics, mortality is very unusual. We deliberately tried to pick a dose that wasn't lethal in mice, but was still significantly paralytogenic. You can see in the black line in panel A about a 10% mortality in virus-treated mice, but those two red curves showing much more increased mortality, decreased survival, are in animals treated with two different models of timing of dexamethasone. And if you look at the animals that didn't die or that you had before they died, and you look at the little red dots and triangles, circles and triangles there, you can see that the clinical disease, not only was the mortality increased, but the clinical disease was worse. So we had IVIG seemed to help, fluoxetine didn't seem to do much of anything, and steroids made things worse. Again, always correlating pretty well with what we saw if we did titers. So you can see there, although the curves look, the dots look sort of closer, that in the steroid-treated animals there was a significantly higher titer of virus in spinal cord. It's hard to know, but of course when you test something in a mouse, you're wondering, does it have any relevance to what we might see in people? We did a really bad study because the gold standard is, of course, a prospective, double-blind, controlled, multi-center trial of a drug. Good luck with that. Can you say herpes simplex and how long it's taken to do all the trials with that? If you take the opposite of all the things I just told you and do that kind of study, it's actually really easy. We asked centers that had treated these children, including our own, to report who got fluoxetine. We looked through the charts and things like that to look at the motor strength testing and so on. And I can just report to you that looking at this kind of retrospective analysis in a non-controlled study, you can see the fluoxetine-treated children on a muscle limb strength score actually fell below the ones, meaning did worse. They were a little sicker, a little weaker at onset, worse at their nadir, and worse at recovery than those who hadn't received fluoxetine. Certainly there was no positive signal here. I wouldn't read too much into the worsening because there was a big selection bias potentially in who got what therapy. But again, does seem like what we're finding in a mouse may also help us predict what would or wouldn't work in people. Many of you may have seen in the Journal of Virology, if you're interested in this, earlier this year, there was again an in vitro study suggesting that telaprevir, this is my one tie-in to the previous hepatitis section, was potentially again efficacious in vitro in inhibiting replication of enterovirus D68, and you can see the target is listed there, the 2A protease. This is from a group at the University of Arizona. You can see the authors there. We're collaborating with them now to test this in mice. We actually, it's a little complicated to give it to mice. You have to do it intraperitoneally, and because these are tiny little baby mice in this model in a corn oil vehicle. But some of the early results are encouraging, although not yet significant. I just sort of put this in to show you not that tenofovir is going to be a good drug or that molecularly engineered variants, which is what we'll actually probably use, will be a good drug, but to say that we're actively screening new compounds. Every time a paper comes out, I get 200 emails from people telling me try vitamin C, try tranexamic acid. We just wrote a paper in JV on tenofovir. And so we're trying to figure out how do you prioritize these, because I think going back to where I started and thinking of the number of cases, this funny every other year epidemiology, the non-predictability of this, it's going to be a really tough row to hoe to figure out how to do this if you only use human data. So the mouse model may be very helpful for us not only to understand pathogenesis, as I showed you, how does this infect neurons, how does it kill neurons, how does it spread, what's its receptor utilization, what are the molecular determinants of virulence, all of which in different pieces and parts we're looking at, but practically and bringing it back to where I started, which is we'd love to come up with something that you could use in humans. So I think, why don't I stop there so I still have some time for questions if people are interested, and thank you very much for having me. Thank you for inviting me.